Good morning, everyone. I am Dr. Rituparna Shaha, Assistant Professor in the Department of Life Sciences, School of Biological and Forensic Sciences at Kristu Jayanti Deemed to be University, Bengaluru, Karnataka. Today, I will be discussing a fascinating topic: amino acids, introduction, and classification. Amino acids are the fundamental building blocks of proteins and play a crucial role in almost every biological process. Let us begin by understanding where they fit in the broader context of biomolecules. To begin with, let us briefly look at biomolecules. Biomolecules are essential organic compounds produced by living organisms. These include carbohydrates, lipids, proteins, and nucleic acids. Each of these molecules has a specific function. For example, carbohydrates provide energy, lipids store energy and form membranes, nucleic acids store genetic information, and proteins perform structural and functional roles. Amino acids are particularly important because they are the building blocks of proteins, which are responsible for catalysis, structure, transport, and regulation in biological systems. Now, let's focus on amino acids. All 20 common amino acids found in proteins are known as alpha amino acids. This means that both the amino group and the carboxyl group are attached to the same central carbon atom, known as the alpha carbon. Each amino acid has an amino group, a carboxyl group, a hydrogen atom, and a variable R group or side chain. This R group is what differentiates one amino acid from another. It can vary in size, structure, charge, and polarity, and it determines the chemical properties and biological function of the amino acid. There are two conventions that are used to identify carbon atoms in amino acids. The first uses Greek letters, where the alpha carbon is the central carbon and the additional carbons in the side chain are labeled as beta, gamma, and so on. The second method involves numbering the carbon atoms, starting from the carbon with the highest priority, typically the carboxyl carbon, which is assigned as carbon 1. Understanding these conventions is important when studying amino acid structure and biochemical reactions. Let us now look at some important properties of amino acids. Most amino acids contain a chiral center, meaning the alpha carbon is attached to 4 different groups. This gives rise to enantiomers or mirror image forms. These enantiomers exhibit optical activity, meaning they can rotate plane polarized light. Amino acids are classified based on their absolute configuration into D and L forms. However, it is important to note that almost all amino acids in biological systems are in the L configuration. This stereochemistry is crucial because enzymes and proteins are highly specific and recognize only particular configurations. Amino acids can be classified in several ways, but one of the most common methods is based on the nature of their side chains or R groups. These classifications include nonpolar hydrophobic amino acids, polar uncharged amino acids, positively charged basic amino acids, and negatively charged acidic amino acids. This classification is important because it determines how amino acids interact with each other and with their environment, particularly in protein folding and function. Non-polar amino acids have hydrophobic side chains. These side chains do not interact with water. Examples include glycine, alanine, valine, leucine, and isoleucine. In proteins, these amino acids are usually found in the interior, where they help stabilize the structure by avoiding contact with water. Aromatic amino acids also have side chains that contain aromatic ring structures. These rings are generally hydrophobic, but some can participate in weak interactions with water due to their structure. Examples include phenylalanine, tyrosine, and tryptophan. In proteins, these amino acids are often located in the interior, where they contribute to structural stability through hydrophobic interactions. However, tyrosine can also be found on the surface due to its polar hydroxyl group. Polar amino acids have side chains that can form hydrogen bonds with water. These amino acids are typically found on the surface of proteins interacting with the aqueous environment. Examples include serine, threonine, and asparagine. Their polarity plays a key role in protein solubility and function. Now, let us look at a special amino acid, cysteine. Cysteine contains a sulfur group, which allows it to form disulfide bonds. When two cysteine molecules oxidize, they form a covalently linked dimer called cysteine. These disulfide bonds are extremely important because they stabilize protein structure, form links with a protein or between different polypeptide chains. They are especially significant in proteins that require strong structural integrity, such as antibodies and keratin. Charged amino acids can be either positively or negatively charged, depending on their side chains. Positively charged or basic amino acids include lysine, arginine, and histidine. Negatively charged or acidic amino acids include aspartic acid and glutamic acid. These amino acids are important in enzyme activity, ionic interactions, and protein stability. Amino acids also have unique acid-base properties. When dissolved in water, amino acids exist as zwitterions, which are dipolar ions carrying both a positive and a negative charge. This happens because the amino group can accept a proton, thus acting as a base, and the carboxyl group can donate a proton, thus acting as an acid. Because of this dual nature, amino acids are also called ampholytes or amphoteric electrolytes. Thus, to summarize, amino acids are the fundamental building blocks of proteins. They contain a central alpha carbon with an amino group, carboxyl group, and a variable side chain. Their properties depend largely on the nature of the R group. They can be classified based on polarity and charge. And they exhibit stereochemistry and exist mainly as L isomers in biological systems. They also act as ampholytes and exist as zwitterions. Understanding amino acids is very essential for studying protein structure, enzyme function, and overall cellular processes. Thank you for your attention. I hope this session has helped you understand the basics of amino acids and their classification.